Mike and I have been working together on GI stromal tumors since 1999 when he walked into my lab and he said, "Have you ever heard of the GI stromal tumor?" And I was one of the few people around who had, and so he said, "Can you get me some?" Meaning pieces left over from surgery. And I said, "Yeah, we can do that." And so we started working together. And I think because we've been working together so long, there's a lot of parallel thinking going on, which means that there's some similarity between his slides and mine. But as we say in medical school, repetition is learning, right? So hopefully this won't get too boring. We'll try to get through it. Do I need to put it on? Okay. Oh, it's recording. Okay. Great. Thank you. So let's go ahead and move forward into the first slide. So Mike presented to you the basic distribution of GIST, 60% presenting in the stomach, 25% in the small bowel, and then we have these other locations. 5,000 new cases per year in the United States. That's a lot when you think about 5,000 individual patients. But compared to colon cancer, 140,000 new cases each year, or lung cancer, over 200,000 each year, it's kind of a small disease. And that's why it hasn't necessarily received as much attention as it needed to in the past. What we see in the microscope as pathologists varies a lot. And that was one of the reasons why originally this tumor was mixed up with a lot of other tumors, because all we had was the microscope to look at. And you can see from these images, all of which are GIST, that they look different. And they get mixed up with other cancers. And that's why the identification of the KIT protein and being able to stain for that allowed us to begin to identify these things much more accurately. And here's an example of, again, that staining. So we're looking at the brown stain for KIT in 3 of the panels. And then there's another marker that we sometimes use called DOG1, D-O-G-1. Interesting story. That was discovered at Stanford. And it was called Discovered on GIST. That's why it was called DOG1. But the real reason is it's because our friend Matt Vanderine at Stanford really loved his dog. True story. Okay. So DOG1 is the other marker. And between these two markers, we can nail down the diagnosis of GIST. So when we first started working in this field, people were still mistaking these tumors for all kinds of other things. Today, I would say we're at 99.9% accuracy here in the U.S. So where did GIST come from? Mike introduced the concept of the interstitial cells of Cajal, the gut pacemaker cells. And this is a cartoon that illustrates for you in yellow on the left this tiny network of cells that lie within the muscular layer surrounding the stomach as well as the rest of the intestine. And there are a lot of similarities between these ICC cells and GIST. They both express KIT, they both express DOG1, they express ETB1, and there are many other parallels as well. So we're pretty convinced that GI stromal tumors come from that population of cells, even though otherwise you wouldn't pay them much mind. But there's even more compelling data. So a number of years ago, and this has now been reproduced by several labs, a mouse model of GIST was made. How was that done? Take a mutant form of KIT, introduce it into the mouse genome, and then now the mouse has an abnormal KIT protein, just like we see in human GIST. And the result of that in the mouse GI tract is an overgrowth of those ICC cells. So all you have to do is change one molecule, make one mutation in it, and suddenly all the ICC cells begin to grow in an abnormal mechanism, in an abnormal pattern. And in fact, they even form nodules, like you see here on the right, down here in the colon, that look a lot like GIST in humans. Okay. All right. So that's pretty compelling data that ICC cells and GIST are related. But where do they come from in the general population? And this was a bit of a surprise, but we noticed early on, and other groups did as well, that occasionally we would run into something that was a GIST, but boy, it was small. And it wasn't something that was picked up clinically, it was incidental. Because the stomach was removed for an ulcer or for a cancer of another type. But nearby, we find a little, like a pebble, like 1 centimeter, less than 1 centimeter in size. Boy, it stains for KIT. It looks like a GIST. And in fact, it is a baby GIST. They even have mutations in them. And how common are these? several studies have now established that in adult population, between 10 and 30% of patients in the general population have one or more of these microgysts in their stomach. So who has one? There's a few more around here in the audience, okay? All right, what that tells us is that there's a lot of these benign gysts in the general population, and most of them never bother us. And there's a parallel here if you think about prostate cancer. There's a lot of prostate cancer in the older male population here in the U.S. Most of it never bothers us. Okay, so if we have several millions of microgysts in the general population, only 5,000 per year actually become something we regard as clinically important, and out of that, maybe 2,000 actually become malignant. Okay. There then are other factors that turn these tumors into something that's malignant. It's not just the KIT mutation or PDGF receptor alpha mutation. That's the starting point that gets those ICC cells growing in an abnormal way and forming these little nodules. But beyond that, we have to have other chromosomal events that allow the tumor to become something much more aggressive and much more dangerous. We have, and other groups have been studying these events. They're beginning to be laid out and understood. And to suffice it to say, that probably takes about 6 steps, or if not more, before something goes from a completely benign gist to an overtly malignant gist. So Mike showed this slide, and I just once more want to emphasize that there is this interesting distribution of mutations in both KIT, and we've been emphasizing KIT because that's the big player. 75% of GIST have mutations in KIT, but also a sister molecule called PDGF receptor alpha. About 10% of GI stromal tumors have mutations in these, and they're mutually exclusive. It's either one or the other or neither. And the neither is the remaining population of what we call wild-type GISTs. Sometimes I like to think of them as wild card GISTs because we used to not understand how these things were growing. Recently, there's been an uncovering of a number of other kinds of mutations hiding in that wild-type population. And so, as Mike emphasized, we're now at the point where we can identify the basic cause for about 97–98% of GISTs. Okay. So Mike also showed how the mutant forms of KIT, or PDGFRA, the dominant players in most GISTs, will cause these signaling events. We talked about those little lightning rods going on. Diagrammatically, or cartoonish, this is what happens going on in terms of that lightning signal being transmitted from the surface of the cell where the KIT molecules acting down to the nucleus, where we see changes in cell, in gene regulation that tell the cell to grow. And by adding a drug like imatinib that goes in and basically blocks that signaling and turns it all off very nicely. Okay. Once more, exon 11 mutant tumors have the best overall response to imatinib and compared to other kinds of GISTs. The exon 9 mutant tumors require a higher dose, and there is an unusual mutation in PDGFRA that we don't have a good drug for except one in clinical trials. Now, the molecular classification of GIST is even more complicated when you begin to look at the wild-type population. This is too small for those of you in the back of the room to read, and I don't want to go through it all. But suffice it to say, there are now a number of other subtypes that constitute this total of 98% that we think we can understand these days. And many of them actually have drugs available. Not imatinib, not sunitinib. It's going to have to be something different. But we do have drugs available for them. One of the most common, though, amongst that wild-type population are this group of tumors called Sdh Deficient Gist. Okay? Sdh is an enzyme. Stands for succinate dehydrogenase. And these tumors are missing that enzyme. They're defective. And they are unusual in that they have a different growth pattern. They're very nodular. And when I see them on the microscope, I think to myself, that might be an Sdh deficient gist. And I can confirm that by doing a stain for Sdh. And if it's clear, there's no brown stain here, that means it's one of these kinds of tumors. They're only seen in the stomach. I don't know why, but they're only seen in the stomach. And they don't respond to imatinib. So we have to think about other treatments for this type of gist. It's about 12% of all GISTs. These are caused by mutations in genes Sdh A, B, C, or D. Those all work together to make that complex of enzymes. And about half of these, we think, are inherited. So suddenly now we're moving from most GIST where you don't have an inherited component to the disease to this subset of GIST where frequently there is an inherited risk called the Carney-Stratakis syndrome. Not only are you at risk for GIST, but you can develop other kinds of tumors like paragangliomas and pancreatic tumors and renal tumors and so forth. Okay. And also, very strange, you can have different penetrants. What does that mean? One patient in a family may have a mutation and develop a GIST. The father may have died of a paraganglioma, and the brother, who also has a mutation, age 60, no tumor at all. We don't know why. So there's a lot of mysteries still that we have in this disease that we need to come to understand. Now, because Mike keeps emphasizing mutations, we got to go looking for them. How do we do that? We do that with what's called DNA sequencing, meaning we're reading the DNA, determining the letters. And remember, DNA is pretty simple. It's made up of 4 letters, A, T, C, or G. And it's the sequence of those that determines what proteins are made and how the cell functions. So in order to look for, in order to read the DNA, we have to first unravel it, and then we create copies of it in the test tube, and that allows us to read it while we're copying it. But every time I see this cartoon, it always reminds me of this cartoon. So the son goes up to mom and he says, so can I go down to the lake? And mom says, well, let me go see what your dad thinks. And he says, well, remind him I could really use some relaxation. So when mom checks with dad, his answer is, if he relaxes anymore, his DNA will begin to unwind. All right. So it kind of reminds me of my brother, but we won't go there. Okay, so we want to copy the DNA, and in the process, we're going to read that. And these days, we've been able to see the benefit of a tremendous revolution in DNA sequencing technology. The original sequencing technology looked like this, a bunch of colored lines going up and down, and the different colors meant the different letters. And you would read it, but it was messy, and it was painfully labor-intensive to create the sequence, and you couldn't read very much. You can only read a small piece at a time. Nowadays, we do sequencing that is basically digital. Out comes the straight, the letters just like this, and you can see the red T's meaning mutation versus the normal A's, and you can even determine quantitatively how much mutation is there versus how much normal there is. Okay. In my mind, this is very similar to the comparison between the TV that we had in my house when I was a kid. Yep, black and white, analog. You had to go over and turn the channel by yourself. Okay. Didn't have a clicker. Okay. We did have one of these, believe me. I'm that old. And today, right? Hundreds of digital channels. Well, our sequencers have millions of digital channels for doing sequencing simultaneously across many different genes at a time. And it's very, very powerful technology. We have actually a couple of different instruments in the lab that allow us to do this. One of them is this box right here. We have a couple of these. And basically, the DNA is spread out on what is essentially a glass slide. Looks like a microscope slide. And then there's a high-resolution camera that's following the sequence. And each of the nucleotides, A, C, T, and G, has a different color. And the camera's just watching the change in color as the DNA is being created. It's very cool. Okay. And it also turns out very large files. So if you think about downloading a movie, this is about 100 movies worth just to do one run of sequencing. The other sequencer, and we have 3 of these in our lab, is one where we do the DNA sequencing literally on the surface of a microprocessor, a little computer chip. Very cool technology again. The DNA is put on the surface of these little spheres. Those spheres are squirted on top of the chip. It sits down, and there's a lot of chemistry going on inside, and from that we can get the sequence out. These are the kinds of things, I have to tell you, these are all only a few years old. This one was first demonstrated to me in 2010, not very long ago. And when the rep came from the company and said, this is our new technology and this is how it works, I said, no way. I didn't believe it. I was like, that's not going to happen. But we've been using it very successfully to sequence GIST and many other tumors. It really does work. we've created a panel of genes. So when we want to really dig deep into a tumor because it doesn't have a straightforward KIT mutation or PDGFR mutation, this is what we go after. We sequence all of these genes simultaneously in one run on one of those chips. And that tells us pretty quickly if there's another kind of mutation, including one of the Sdh genes, that we want to know about. Now I want to show you, in a sense, what it's like then to try to interpret DNA sequence, okay? And we're going to use an example from the Bible, all right? I hope this is visible to you. There is this segment of a Bible that was published in the 1600s, King James Version, and it says here, "Thou shalt not kill, thou shalt not steal, thou shalt not bear false witness." Sound pretty familiar? There's one problem. Right here it says, "Thou shalt commit adultery." Whoops. Whoops, wait a minute. That's not supposed to happen, okay? So this was known as the Wicked Bible, 1632 edition. Once the error was discovered, they went around the countryside trying to grab all those editions and burn them. But as it turns out, some of them survived, and we have this as an example of a printing error, right? What happened was there's a deletion of one word, the word not, and we see that exact same thing in the DNA sequence of GS stromal tumors, okay? Here's the KIT gene. We're looking at a series of amino acids which are encoded by these nucleotides from the DNA, and right here there are 3 of them missing, which means that aspartic acid in the KIT protein is gone, and that's all you need. That single change turns the KIT molecule on and makes it a very powerful driver for the growth, the initial growth of a GS stromal tumor. One more example is another one from the Bible, 1795 edition. We have, "Let the children first be filled," became, "Let the children first be killed." Ouch. Okay. So that is an example not of a dilution, but of a substitution, right? We substituted one letter for another. We see exactly that thing in KIT as well as in other tumors, the BRAF gene. We're replacing this T with an A, and that's all you need to do to go from valine to glutamate, and that means this is turned on and it's driving the growth of a melanoma in this case, but we see similar mutations in KIT as well. Okay, so it gives you an idea of the kinds of changes, very subtle changes in the DNA code that lead to these very considerable consequences that we see in tumor growth. Okay, now Mike wanted me to discuss a little bit about how we assess how actively growing a tumor is and what the prognosis is. Okay. First, I want to show you this study that was done prospectively, meaning from this period forward for a period of time, we're going to see in this particular region of France, the Rhone-Alpes region of France, how many GISTs are diagnosed. And this was over a 2-year period, and the answer was 115 GISTs were diagnosed. Okay, so now you're following it, not looking backwards in time, but just in real time over a 2-year period. How many new tumors? 115, and out of those, 88% had not spread. So that's kind of good news, because those 88% of patients theoretically are cured by surgery. Okay, only 12% actually had metastatic disease when they walked in the door. Okay, so that's great. However, we then need to look at the risk of recurrence, and I'll show you how we do that in a moment. But the risk, as you see, varies considerably from about a third or a little bit more than a third that are very low risk, probably don't need to take Gleevec at all, to some that are in the middle region, and then more than a quarter that are high risk, and they definitely need to be taking Gleevec. Okay, so what that means is that even though many of these patients are going to be cured by surgery, a considerable fraction are going to have to consider the use of Gleevec after the surgery. So how do we assess it? Mike introduced 4 factors that we look at: location, size, and mitotic index. Sorry, 3 main factors, tumor rupture being the 4th one. The 3 main factors, and of those, mitotic index is the most important. Now, what does that mean? What is a mitotic index? This is a diagram reminding you from your biology days in high school, okay, where we're taking a cell through cell division and dividing it up into two new daughter cells. And you'll remember that during that process, the chromosomes have been replicated, and at one point they all get lined up because you've got you get one copy of each on each side, and they get pulled apart into the daughter cells. This is called mitosis. This process is dividing the chromosomes up into the two new cells. That's mitosis. Okay? So we can look for mitosis on the microscope. And if we look at very, very high power, this is what it looks like. So that's exactly the diagram I showed you just a moment ago, right here, like that. That's what we're seeing on the microscope right here. Okay. Now, if we go at lower power, because I don't want to spend all day looking at that, that's kind of like trying to mow, a gigantic lawn with a teeny little lawnmower. So instead, I'm going to back off to a lower power, and I still can see them. And there they are at lower power. Here you can see them dividing up chromosomes, and these ones are still lined up together. And so these are examples of the mitotic figures that we look for. And of course, by definition, when we see those are dividing cells. The more we see, the more the tumor is dividing. Very simple. So all I have to do is cruise around at this power on my microscope, about 25 different areas of the tumor, and then add up all of the mitoses that I see. Hopefully it's only one or two, but if it's more than 5, it's a sign that there could be a problem and that this tumor might come back. So we combine all this information together. The mitotic index, by far the most important. But we also factor in the size of the tumor, if it's 2 or 2 to 5, or it's greater than 5 centimeters or greater than 10. And then the location. Mike talked about how the gastric tumors have a better prognosis overall than other tumors, and that's borne out in these statistics here. These, by the way, this was put together by a couple of pathologists working back in Washington, D.C., from old cases before Gleevec was invented. And it was a heroic effort. This is based on over 1,000 patients with long-term follow-up. And by getting all that information together, we can generate this chart, and that's what allows us to fairly accurately predict what is the risk for a patient, and they can decide whether to take the drug. Okay. So just to summarize then, what we've talked about is that GIST are a family of tumors arising from mutations in a number of different genes, not just KIT. Most GISTs probably come from these little microtumors that are common in the population, but you have to have other kinds of events genetically that allow them to become malignant. We talked about how next-generation sequencing is very helpful if we're going to do deep molecular subtyping of these tumors, identifying unusual mutations. And finally, how our mitotic index is the most important, but we have these other factors that we need to look at in order to judge whether the tumor is going to come back. So with that, I'm going to end, and I'll turn the podium over to Tracy.